Good morning, everybody. And I believe the next thing on our agenda is Dr. Planis's presentation, but I believe Steve has some logistical announcements first.
Good morning, everyone. Welcome to day 2. Just a reminder for those here in the room, if you do want to sign up for public questions or comment, the sign up is right out there at the registration desk. And of course, for the web audience, we'll take yours in the question pane in the webinar. Obviously, you all figured out that we're meeting in this room this morning. When this morning's session is complete, we'll ask everybody to leave the room as quickly as they can because they're going to make a room over for the conference board, which begins this afternoon in this space. The commission meeting will continue for the afternoon in the space next door, the aft deck. And the processor advisory board begins their meeting this afternoon in the Endeavor room. And that'll be the room arrangement as well for tomorrow morning. I'd like to remind everybody that the IPHC reception is tonight at 7 o'clock. It's in the quarter deck, which is at the top of Tower 1. So don't miss the IPHC reception tonight. And if you have any questions during the course of the meeting about the logistics or anything else, or you need any help with anything, again, the reminder that the Secretariat staff is at the back of the room, right up here. Thank you.
All right. Thank you, Steve. And so we'll jump right into Dr. Planis's presentation on the IPHC 5-year research program. Dr. Planis.
Thank you, Commissioner. Good morning, Commissioners. I'd like to present to you today an update on the biological research activities that the Biological and System Science branch is conducting. The outline of the presentation is we're going to start with a reminder of the 5-year research plan and the management implications, and then I'll dwell into current progress on ongoing research projects, and I'll highlight some of them. I will not cover the entire research activities. And finally, I'll make a mention of externally funded collaborative research. So the 5-year Biological and System Science research plan contemplates work on 5 primary research areas. The first one is indicated here on the top left, migration. The second one is reproduction. The third one is growth. The fourth one is discard mortality rates and discard survival. And the fifth one is genetics and genomics. The first area, migration, has as its main objective to improve our current understanding of migration throughout all life stages, larval, juvenile, adult feeding, and reproductive migrations. And the management implications are obviously in stock distribution and regional management. The research area on reproduction, the main objective is provide information on sex ratios of the commercial landings and to improve current maturity estimates. The management implications are in our ability to estimate the female stock spawning biomass. The third key research area, growth, has as its main objective to improve our understanding of factors that are responsible for the historical changes in size and age and the development of tools for our ability to monitor growth and physiological condition in Pacific halibut. And the management implications of this work are in our ability to correctly estimate biomass. The fourth key research area, discard mortality rates and discard survival, obviously as its main objective is to improve our current estimates of DMRs in the directed longline fishery and to provide new and experimentally derived DMRs in the guided recreational fishery. The management implications are in our ability to correctly estimate discard mortality. And the fifth and last research area, genetics and genomics, has as its main objective to improve our understanding of the genetic structure of the population, create genomic tools, namely the genome, and to use genetics in order to further our understanding of migration and distribution as well. The management implications are in stock distribution and local adaptation of the fish population, of the Pacific halibut population. The biological and system science research activities highlighted here in the center of this diagram in red are dictated by the needs of stock assessment and the MSC process, but also through our need to provide basic and applied understanding of Pacific Health biology. The IPHC subsidiary bodies, in particular the Science Review Board and the Research Advisory Board, they both provide important inputs to the biological and ecosystem science research program in the way of review and direction in terms of research activities. And the IPHC subsidiary bodies then recommend particular research activities to the commissioners. One of the things that we strive on is to integrate biological research activities with the needs of stock assessment, management, strategy, evaluation, and policy. And in this table, which you probably cannot read the final or the small text, is just to highlight that in every single research area, there are several research outcomes that are identified and that we're working towards because they are relevant for stock assessment, highlighted in green, and also that provide important inputs into both stock assessment and the MSC process, as shown in blue in the last column. Just to give you an example of what we mean by this, for instance, the work that we're currently conducting on identifying sex in the commercial landings, providing sex ratio information of commercial landings, represents an input, which is sex ratio at age, that is used in stock assessment, as you saw yesterday in Dr. Stewart's presentation, providing spawning biomass scale and trend, but is also an important input in the MSC process in the operating model. This research activities are actually temporarily identified through the existence of this 5-year research plan, and I'm just providing you here an example of 3 areas and how those activities are done or programmed through time. The first one is migration, and you see here boxes in orange that are the data synthesis, and that's where we actually try to incorporate this biological data coming from this research and make it applicable to stock assessment and MSC process. And the same can be said for other areas, reproduction, for instance, and growth. Reproduction probably being the first one where we provided already a direct input into stock assessment, as you saw yesterday, with the sex ratio information. So let me move on with describing our current progress on some of the ongoing research projects. I'm going to start with the first research area, which is migration and distribution. And currently we're undertaking 2 specific projects. One is to look at larval and early juvenile dispersal, and the second one is to look at late juvenile and adult migration. The first one, larval and early juvenile dispersal, has as its main objectives to describe the contribution of specific spawning grounds to settlement grounds. More specifically, to look at the connectivity between different ocean basins, for instance, the Gulf of Alaska and the Bering Sea, and to look at environmental effects on larval distribution. These studies are done in collaboration with the Alaska Fisheries Science Center in Seattle, and are conducted using data that is derived from the ichthyoplankton surveys that NOAA conducts. So we have information on larval abundance and distribution, but also we're doing considerable amount of work in modeling potential larval dispersal pathways. So we're looking at from specific spawning grounds where larvae could actually end up going. And this is what you see in this little small graphs in the left-hand side of the screen. So we can actually model larval trajectories based on where those larvae are spawned. I'll explain that in a little more detail in the next slide. But also we're looking at dispersal of young fish post-settlement. So we're looking at using Dr. Webster's space-time model. We're looking at changes in abundance and distribution of fish that are 2 to 3 years old, and we can actually follow specific year classes. And in this example, you see on the top age 2 fish and how they're concentrated, yellow meaning higher abundance, and how those fish at age 3 are actually starting to move south from the eastern Bering Sea and almost into the also into the Gulf of Alaska. And this work is in progress right now. So related to larval dispersal, I just wanted to show you this slide in which we model the outcome of spawning from different areas in the Gulf of Alaska and the Bering Sea. So on the top left graph, you see 5 different spawning locations, one being in the Bering Sea, and then 2, 3, 4, and 5 in different colors in the Gulf of Alaska. And we model the productivity of this spawning areas in terms of where those larvae will end up. And the map that you see below that one, on the lower left side of the screen, you see the different areas in the Gulf of Alaska and the Bering Sea where this modeling effort is taking place. So for instance, just to give you an example, if you look at the right-hand side of the screen, we have the first 3 spawning areas, spawning area 1 in orange, spawning area 2 in light green, and spawning area 3 in bright green. And what you see here with this different heat map is that the spawning area 1, which corresponds to the Bering Sea, in fact, does not produce larvae that are modeled into the Gulf of Alaska. And almost 100% of the larvae that are produced by that spawning area remain within the Bering Sea. Interestingly, if you look at spawning area 2, which is the next one to the right, and that's in the Gulf of Alaska, obviously a great proportion of those larvae will remain within the Gulf of Alaska, about 50%, but the other 50% will actually most likely go into the Bering Sea. So there's a potential important contribution of Gulf of Alaska larvae into the Bering Sea. And even spawning area 3, if you go east, then that spawning area produces probably 80% of the larvae that will end up in the Gulf of Alaska, but about 17 to 20%, those larvae will end up also in the Bering Sea. So there's we're looking at important potential contributions of Gulf of Alaska fish into the Bering Sea, and that's one of the examples of that connectivity between ocean basins that we're currently studying. To move on to late juvenile adult dispersal, we're doing tagging work, and this is work that has been conducted by the Commission for many, many years. We're conducting wire tagging of under 32 fish, both in our own Fisheries Independent Setline Survey as well as in the NIMS Trawl Survey. Since 2015, we've tagged almost 11,000 fish, of which we've had already 132 recoveries. This is obviously a long-term project. And in this map, you see the different, all the dots corresponding to the fish stations, and the fish that have been recovered so far. So in the triangles is the recovery location, and you see that there's a fair amount of movement, obviously, as we know. We also use electronic tagging to understand early adult and reproductive migrations. And in 2019, we focused our efforts, in fact, in the northeast Bering Sea shelf, in order to start looking at potential connectivity between the eastern and the western North Pacific Ocean. We actually did a project in collaboration with the Norton Sound Economic Development Corporation and the University of Alaska Fairbanks, and we tagged about 15 fish from St. Lawrence Island, 24 fish from Norton Sound, and 5 fish from the on the NIMS Trawl Northern Bering Sea Trawl Survey. And this data are actually coming in as we speak. So that will help us understand much better the level of connectivity between the East and the West North Pacific. And this is a key area of interest for the Commission, as Dr. Wilson highlighted yesterday. And in fact, as he mentioned, in order to pursue potential collaborations with Russian, Japanese, and Korean scientists that are invested in our shared resource, we organized a Pacific Halibut workshop within the Pisces meeting in 2019 in October, and that was highly successful. We had participation from Japanese, Russian, Canadian, U.S. scientists. And in fact, there's going to be a second Pacific Halibut workshop in this year's Pisces annual meeting, and we'll be in China, and that will be to be seen whether travel bans are going to be in place or not. The second key research area is that of reproduction. We have 2 projects. The first one you've heard considerably about. It's the sex ratio of the commercial landings. The second one is the full characterization of the annual reproductive cycle in order to improve current estimates of maturity. The first project, I'm not going to go into the details. You've probably heard this before. We now have genetic assays that can identify males from females using a single nucleotide polymorphisms, and that are used in our lab that I'll describe in a minute, and with high accuracy. So as you heard yesterday, we have just completed the entire set of aged 2017 and 2018 commercial samples, more than 10,000 fish per each of those years. And in those 2017 and 2018 sex ratios have been used in the 2019 stock assessment, as you heard yesterday. So this is work that is actually being conducted in our office in Seattle. It's as a result of the laboratory that we established in 2018, and Ms. Anna Simeon has been key in developing this assays in the lab. Those assays were developed in collaboration with the University of Washington, but now we're been implementing those in the lab. And Ms. Simeon was supported, well, is still supported partially by North Pacific Research Board grant until March of this year, and then she's now continuing with us for another 2 years. In addition to the sex ratio information, we actually use the lab for many other projects. So this laboratory is actually key to ensuring our progress in the other research areas, growth and reproduction, discard survival, and importantly, because it's a site where we could actually train staff and students in specific techniques. And in particular, earlier this year, we've had a master student from Alaska Pacific University, Ms. Teresa Fish. We've had the 2019 IPHC intern participating in laboratory activities, and we've also had a high school senior doing his senior project in the laboratory as well. The second project is the full characterization of the annual reproductive cycle, and the main objective here is to revise maturity estimates for female Pacific halibut. And as you probably know, maturity is estimated visually in fish that are sampled in our fisheries independent satellite survey. So it's a macroscopic maturity staging, and fish are classified in terms of their level of maturity according to 4 classification criteria: immature, maturing, ripe, and resting. And Dr. Stewart considers maturing, ripe, resting as mature. But this is obviously a visual identification of a maturity status, just looking at the condition of the gonads. And we want to know whether this is entirely accurate or not. So the way that we're approaching this is by doing a histological assessment, by looking at microscopic maturity staging, which accurately provides the best information possible on the developmental stage of the ovary that is collected. So through histology, we do an oocyte stage classification, and based on the developmental stage of the oocyte, then we stage those females. So eventually, this is work in progress, we'll be able to compare the maturity estimates that are currently provided by this visual maturity staging versus the new ones, and we'll see whether the current maturity maturity at age is still holds, and whether it needs to be changed. So this study that I've just described was conducted between 2017 and 18 in terms of the collection of the samples in one particular area, in the Portlock area in the central Gulf of Alaska. Now we're generating a tremendous amount of data related to those samples. For instance, in the top right, you see a graph in which the x-axis is months from January to December, from left to right, and the different maturity classifications. Just look at the pink bars, and you see the pink bars increasing in size at about July and August. That's when you start seeing more and more females in that maturing stage. And below is the gonadosomatic index. That's basically the proportion of the ovarian weight relative to the round weight of the fish. just gives you an idea of how much the gonad is actually growing. And there's a fair relationship between those two parameters. Well, what's interesting is that we obviously collect for our maturity estimations. Currently, we collect those samples within the FIS. And as a reminder, that collection takes place between late May, early June until early September. And that's precisely the time when we're actually start to see more and more females moving into that maturing category. So what we propose for this year actually is to start looking at potential spatial changes in maturity schedule. So we're looking at the sampling coastwide for maturity and conducting histological analysis and looking at potential differences in maturity schedules throughout the coastwide collection. And we're looking at collecting samples in the FIS either in July or August, which is we consider an appropriate time. The third area is growth. I won't dwell on this area particularly because you heard it last year. There's a manuscript that is now being prepared for publication, and the details are actually outside. There's you probably noticed that there's a few posters that are highlighting some of our research with quite a bit of detail. And we'll be happy to show more detail on specific projects if you wish. So please grab us during breaks and at any time for a little more information than I'm providing here right now. But the work on growth is obviously related to trying to understand those historical changes of size at age that Dr. Stewart was discussing yesterday. And we're looking at obviously potential changes in growth as one of the factors, one of the many factors possibly responsible for this historical changes in size at age. But we need to understand growth in Pacific halibut, and we need to develop tools to measure it. So the first project is really the identification and validation of physiological markers for growth. If we could take a sample of a fish and we can tell whether that fish is in a positive growth trajectory or a negative growth trajectory, that's going to be extremely useful information to understand the growth patterns. And the next step is obviously to evaluate growth patterns in the Pacific halibut population and to look at possible effects of the environment on growth. So this is work that is being conducted, partially funded by a grant from the North Pacific Research Board and in collaboration with the Alaska Fisheries Science Center in Newport. And again, details are outside in those posters, and I'll be happy to explain those to you. The fourth research area is discard mortality rates and survival assessment. And we have two specific projects. You've heard about the first one, the one on the directed longline fishery, and the second one is a new one that is on the guided recreational fishery. The first project on providing direct estimations of discard mortality rates in the longline, directed longline fishery has had many objectives, one of which was to establish a relationship between handling practices and injury levels and physiological condition of the released fish, and ultimately linking it with survival. So one of the first data that we generated was this graph that you see on the right-hand side, where on the x-axis you see 4 different release conditions from left to right: excellent, moderate, poor, and dead. And then the different bars and different colors represent the different release methods. So in green you have careful shake, in blue you have ganging cutting, and in red you have hook stripping. And what you can see here is that the first 2 release methods, careful shake and gang and cutting, produce the least amount of injuries, as everybody knows. But we want to link this information with also the physiological condition of the released fish. We want to know those fish, what are their stress levels, and whether that impinges a burden on the survivability of those fish. And we're looking at, specifically, we're looking at blood stress indicators, such as metabolites, glucose, lactate, or stress hormones, such as cortisol. And here I'll present preliminary results that we have on the first 2 determinations on glucose and lactate levels in the blood. And here you see on the x-axis again the release conditions of those fish from dead, excellent, moderate, and poor from left to right. And on the y-axis you see the levels of glucose and lactate. And I'll just highlight the first category, those fish that are actually classified as dead. Those are the fish that have the lowest glucose levels and the highest lactate levels. So the idea is to look at some of these physiological indicators and see which ones or which combination of them can be useful predictors of survivability. So this is work that's still in progress. The second project is related to the establishing for the first time direct estimates of discard mortality rates in the guided recreational fishery. This project was initiated last year. The first phase was basically just to collect information on hook types and sizes and handling practices. So in 4 ports, 2 in regulatory area 2C, and specifically in Sitka and Juneau, and then 2 in regulatory area 3A, Homer and Seward. We conducted dockside interviews and we collected, again, information on practices and gear. In order to design the experiments that are going to be hopefully conducted this year in 2020, where we are going to be investigating the relationship between gear types and capture conditions and the size composition of the capture fish, and also the survivability with injury profiles. So we're going to do a similar experiment that we just conducted on the directed longline fishery with the use of accelerometer tags that provide very good information on the survivability of those fish once they're released. And hopefully we will be able to provide direct estimates for the first time of DMRs in this fishery. This work has been done with the support of a grant from the National Fish and Wildlife Foundation that IPHC leads, but in collaboration with the University of Alaska Fairbanks, Alaska Pacific University, and also the Alaska Charter Association. Finally, the last research area is genetics and genomics, and we have 2 projects. One is the establishment of the genetic structure of the Pacific halibut population and use of genetic information to better understand distribution. And the second one is genome sequencing. I will only speak to the first one. And in order to conduct the studies, we've hired a new biological position at IPHC, Mr. Andy Jasonowicz, initially for a 1-year contract. We hope to extend that for a little more. And he's going to be crucial for our ability to move into the application of genetics to understand stock structure and distribution and movement. So the first project that we are undertaking is to look at regulatory area 4B structure. And this is based on some preliminary information that we generated some years ago, and that there appear to be some genetic differences between fish on either side of Anchorage Pass. So fish from between Western and Central Aleutians. In fact, those results are in one of the posters that are outside. So the aim is really to investigate potential genetic differences between Central and Western Aleutian Islands. And in fact, we're collecting, as we speak, winter genetic samples in Attu and in Adak, with which we will conduct genetic analysis. The second project is the identification of genetic or potential genetic signatures of origin, what is referred to as baseline signals. The aim of this study is to establish those genetic signatures or genetic baselines from known spawning groups to conduct assignment studies and determine the genetic population structure coast-wide. So we're using samples that are being collected right now in Central and Western Aleutians. We're using winter samples that were collected in 2018 as part of the reproductive assessment project. And we're also going to be using some historical samples that we have and that were used in that previous study, including samples from Russia. So this is work that has been in the planning stage right now, and we hope to start as soon as we complete the full set of winter samples for genetic studies. So let me just finish by highlighting that in addition to the projects that you've heard that are IPHC led, those are the ones where IPHC appears in blue, we collaborated in 2019 in 2 studies that were not led by IPHC, but demonstrate, the interest in collaborative research, specifically the first one that was in collaboration with Alpha, and that was related to whale depredation and whale detection. And the second one was in collaboration with Pacific States Marine Fisheries Commission, and that was related to the use of LED lights in trawl nets to reduce Pacific halibut bycatch. So those two studies were completed and finalized, and we're in fact engaging in further studies with these groups for the coming year. And that completes my presentation. Thank you.
Thank you, Dr. Planas. We'll pause and see if there are any questions from commissioners. Seeing none, I understand you're also going to provide report number 8 on the Research Advisory Board.
Correct.
But just a note to the public, because we got a little off schedule last night, any public comment on item 7 that Dr. Planas just presented, we'll roll that into the next public comment session later this morning. Well, go ahead, Dr. Planas, with item 8.
Thank you, Commissioner. Let me proceed with the report of the 20th session of the IPHC Research Advisory Board, RAP 020. This meeting took place February 27th in 2019. And I'll just go straight into the recommendations that resulted from that meeting. One of the recommendations was related to the IPHC closed area, and the RAP recommended that in coordination with the North Pacific Management Fisheries Council, that the IPHC Secretary examine alternative management regimes for the closed area, and for these to be presented at the 96th session of the IPHC annual meeting. There were a few requests. One of them was the effects of longline gear on benthic habitats, lost gear, and spatial patterns in fishing. And the request was that the IPHC consider research examining the following aspects of the longline fishery. A, the impact of longline gear on the ocean bottom. B, the magnitude and impact of lost and abandoned longline gear over time. And C, the extent of the geographic footprint of longline gear. Second request was related to black cod pot fishing. And the RAP requested that IPHC gather data on the effect of this shift, including potentially, A, how this change affects the catch of Pacific halibut in the sablefish fishery, B, how the gear shift in the sablefish fishery might drive whale predation toward Pacific halibut fishery, and C, the change in this effects over time. The third request was related to the impact of recreational fishery releases, and the RAP requested that the IPHC Secretariat begin to explore such research possibilities, including guidance and best practices that might be required. A second request related to the recreational fishery releases was that the RAP requested that the IPHC Secretariat work with the relevant contracted party agencies to encourage and enforce complete data collection. And that concludes the set of recommendations and requests from the RAP in the last meeting, and those will be addressed in the upcoming RAP meeting that will take place in Seattle on February 26th of this year, later this month. Thank you, Commissioner.
Any questions from commissioners on that report? Mr. Ryall.
Yeah, thanks very much. Dr. Planas, on the requests, so you said these were coming back to the meeting this year. Does that understand? Is that what you were saying? And if so, how will they be handled? What will be the outcome?
Thank you, Commissioner. Through the chair, Yeah, this series of requests and recommendations will be addressed and will be included in the agenda to the meeting. Documents have been produced to provide some information already. The documents for the RAP, upcoming RAP meeting 2021 will be, the RAP 021 were published in January with information related to all these items. So this information can actually be found in documents that are already published.
Excuse me. Okay, thank you. Thank you, Dr. Planas.
Thank you.
I think our next report, number 9, is from Dr. Sean Cox on the Scientific Review Board.
Thank you, Commissioners. So every year I come and give a little overview of what we've been doing in the Scientific Review Board. Just remind you of the membership of that board. There's myself, Dr. Mark Mangle, who is now retired. I assume there might be some work on finding a replacement for him fairly soon. Dr. Jimmy Anelli, who's from NOAA. Kim Scribner, who's from Michigan. He is a population biologist. And a new member, Dr. Sven Kupchus, who's from the UK. So we had 7 recommendations and 4 requests this year. I'm going to go over a few of them here. And then in the second part of the talk, I only have 15 minutes, so I might go fairly quickly. But I wanted to just give you an overview of what peer review means to the SRB and then what our process is for actually conducting these reviews. So one of the first recommendations was basically to, as you might have seen yesterday from Dr. Stewart, some review of the impacts of bycatch on directed yield. And so that's an effective tool to use to get sort of the relative impact of bycatch on directed fisheries over time. But there's a lot of factors that go into it. So when you see kind of one trend line or a set of estimates in what the effect might be, you know, there's probably a dozen things that could affect those particular lines. So it really, you know, how you calculate bycatch and what you do with that information really depends on how it's going to be used in management. And so that leads to this recommendation, which is that if bycatch management is actually a critical issue, which I assume it probably is, then that's the management strategy evaluation is a good place to incorporate that, because like I said, there's so many factors that go into it. The MSE part is where you can kind of play those scenarios out and test different bycatch management strategies, because it's not a straightforward thing that that bycatch impacts have on directed fisheries. And I think we've said this before as well, that certain things like replay analyses, you know, they can be effective to give you an idea of what might have happened in one particular instance, but really, what if questions belong in the management strategy evaluation work. This year there was an independent external peer review of the stock assessment, and so we were encouraged by that review. And one of the things we wanted to highlight was the external reviewer had an in-person some time actually at the commission, at the secretariat, to familiarize himself with the data, with the personnel, with the system that's actually used in the science. And so we strongly endorse maybe formalizing that in-person review component in future external reviews, just so that it's done effectively. And when SRB was first created, we actually spent about 3 days digging deep into all the data and all the methods and historical methods and things that were used. So it's a really effective way to get somebody up to speed. So that you don't get a review that doesn't really, you know, that makes a lot of errors in assumptions. This is work that we're recommending for the upcoming research meeting in June, mainly looking at some of the more technical details of the stock assessment. And some of the things on management strategy evaluation, we noted here that there was an objective to have annual mortality limits related to local abundances. And so we wanted to point out here that that may or may not actually be relevant to other objectives. For instance, trying to maximize yield in a particular area, you may not want mortality limits related to local abundance. There may be other ways of doing it that actually work better. So in this case, what we would do is recommend that these kinds of objectives be translated into management procedures. So for example, one thing you could have is fixed, I don't know what word I want to use here, but fixed distribution of catch among regulatory areas might actually work better than having those things fluctuate over time in related to local abundance. Those are the things that the MSE should be testing rather than trying to achieve. And this is kind of related to that, where we're requesting more to the MSAB that they define objectives independently of management procedures, and that's kind of what I was getting at in the previous one. Try to define the things that you're trying to avoid or achieve, and then go to the task of finding the methods that you're going to use to get there. Those are separate issues. And this one here is basically recommending that the commission develop a standard criterion that should be achieved for that any management procedure that's being considered in the MSE should be able to achieve a certain criterion. So this is used in other RFMOs, in particular for southern bluefin tuna, for instance, where there's a standard criterion, which is that particular stock, if you don't know about it, it was severely overfished and has been rebuilding from about 5% of its unfished abundance. So it's got a long rebuilding process that's underway. And they decided to create a standard criterion that 30 years from now, any management procedure needs to be able to have the stock at, I believe it was 30% of its unfished abundance within 30 years with a 50% probability. So that serves as a filter so that no management procedure that can't meet that criterion goes any further. This really helps to reduce the dimensionality of some of these problems, because trying to rebuild that stock at that rate to hit that target actually creates a big constraint on other things. So it helps to reduce the dimensions of the decision making. And it's consistent with other RFMOs, which I know is also important. Yeah. Okay. So that leaves me a few minutes to talk about peer review, and the SRB meetings are webcast. I believe recorded or live. I think they're live, but we have very little in-camera private discussion, only at a few times while we're deliberating on the report. But for the most part, these are open that anybody can actually see these. So I wanted to define here what peer review means in the SRB context, and then what our particular process is. So there's different types of peer review. I don't know if you guys are familiar with them, but there's scholarly peer review. This is the 350-year-old process of vetting scientific journal articles and new discoveries and things like that. The SRB peer review is not about scholarly review. Scientific journals have different standards than we're looking for. Scholarly reviewers have different expertise that may not be specific to this context. And especially scholarly reviewers, when you submit a paper to a scientific journal, they could reject it outright. And that's not really something that's open to the SRB. We don't really ever contemplate rejecting a stock assessment. that would, I mean, the iterative nature of the process is kind of works against that. There's professional peer review, which is things like clinical trials that's used in medicine. It's mainly to try to ensure compliance with broad scale standards and accreditation. This is also not what we do in SRB, particularly because fishery science and stock assessment doesn't have commonly accepted standards. It's not actually, engineering and other things that have standards to work on. And we have no input on any kind of promotional issues within the commission secretariat. Technical peer review is mainly used in engineering to comply with specific statements of work, technical specifications and safety issues. This is a little bit different from the one I mentioned a minute ago. In this case, engineers want to build something like a bridge. There's a statement of what the bridge should be able to hold and things like that, and then the engineers go out and deliver on it. So I looked into this one because this is actually more like what we do, and so in looking into the NASTA Systems Engineering Handbook, which when you want to talk about complex systems engineering, this would be the place to look. Peer reviews, inspections are a well-defined process for finding and fixing defects. And that would be, in a nutshell, what the SRB is actually trying to do. And in particular, it's conducted by peers that have assigned roles or specific areas of expertise, and also with a vested interest in the work product that's under review. So in the SRB, as I mentioned when I went through the members, we have members who are not explicitly assigned to cover an area, but we do cover a bunch of areas. So we have expertise in survey design and analysis, stock assessment methods, management strategy evaluation, fish population biology, and so on. So we do cover pretty much everything that's done in a stock assessment. Peer review inspections are also done within development phases and milestones, and this is quite common in the SRB, that there are things that are coming down the road that we provide advice on in phases along the way. So for instance, some of the that Dr. Planis was just mentioning, some of the sex composition of the catch, those kinds of things came up much earlier in the SRB, and we discussed options for going forward with that and reviewing some of the initial pilot work that was coming out. Survey design is another one. That's one of the more recent topics. So technical review is really the closest to what we do. Some of the key differences, though, are the SRB reports to a bilateral commission, whereas a lot of systems engineering technical reviews respond to unilateral kind of private interest in private industry or in government, where the goals and objectives are clear. And especially the statements of work are quite clear in these other contexts. We don't really have a very specific statement of work that we work from. What we generally do is review progress against requests that we've made in the past, as well as things that staff bring up that they have questions on, that they want some feedback on. So we're more of an iterative process than a rigid statement of work type approach. In technical reviews, reviewers typically have a vested interest in the work product. In particular, they represent areas of the life cycle or the process that could be affected by what's being reviewed. And this is kind of interesting. Basically, that we need to have expertise in these different areas that are under development, and then at least some, and this is sort of my addition to this, is that some should have some skin in the game. So in particular, that if errors are made in a stock assessment, that somebody is aware of the downstream consequences that might have. And so people who don't have skin in the game, so if you had a bunch of academics like me alone doing your review, we don't really have skin in the game, so we could make a lot of recommendations that may have unintended consequences. So in the SRB right now, we have typically, historically, we've had 2 to 3 academics plus 1 government scientist, who is Jimmy Anelli. now we're with Mark leaving, we have 2 academics and 2 government scientists, although Sven really doesn't deal with any fisheries that are affected by the science that's produced here. But in any case, it kind of keeps people more honest that way, and not that they wouldn't be, but it definitely helps to have some skin in the game. Okay, and then just to wrap up here would be the process. So we typically, well, not typically, but I think it's by design, we get background materials up to 30 days prior to the meeting. And so those would be documents documenting progress on the requests that we made in the past, proposing new topics, and some things that staff are requesting for advice on. And quite often the staff will draft recommendations that they hope we can decide on in the meeting. These may or may not stand or turn out the same in the end. And then at these meetings, so we meet in September where we talk more about the tactical issues involved in the stock assessment. And in June, which is more of an open research, long-term research topic meeting. And so we review inputs first from the party reps. So US and Canada have representatives that take comments, questions from you or from the commissioners. And we deal with those at first. And then we go through a series of presentations on different topics for the different areas that are involved. And then, as I mentioned earlier, we do have some in-camera drafting sessions where we try to push the issues where we can. And so it's good to have those in camera because sometimes you work through some issues that, it's kind of, you don't want to see how the sausages are made. And so we draft some requests and recommendations and things like that. And rather than just walk away and kind of hand these recommendations and requests to the staff, we actually invite them in and we all sit down and talk about them. So, because some of the requests might not be feasible, we might have made an assumption that was incorrect. So we clarify everything so that when they're sent off to address these requests, that they know exactly what they're going to do and how we'd like it reported. And then finally, fairly quickly, we go through some quick revisions and magically it appears on the website. And then this process is kind of iterative and goes around and around. So with that, I'll take any questions. I might have missed a few things on process. I don't know if anybody has any questions.
Mr. Ryall.
Thanks very much, Dr. Cox, and for the extensive history around peer reviews. I had a question on slide A6 on the biannual peer review that you were proposing. I think it's a good idea to do that, but it seems like doing it every 2 years seems to be fairly frequent. And so why every 2 years? And then I have a more lengthy question, I suppose, and I was considering asking Dr. Planis as well about RAB. Both of these bodies have been in place for a number of years, and as you noted, there was an extensive external peer review last year that focused on a couple of critical areas. But I guess my question for you, and would be maybe if Dr. Planis wants to go up and answer too, is how do you view that the SRB and RAB is working? As you were going through your recommendations, a lot of them are very technical and focused towards, a number of them were focused towards the MSE process, and we'll hear from Dr. Hicks shortly on that, and also from the co-chairs. But I guess, as you outlined when you went through the process, there's a lot of back and forth with government scientists and also with the MSE Secretariat staff. And so going back to my question, how do you see that the process is working from your point of view? And when these recommendations come to the commission here at your last slide 26 says the commission would note the reports for the scientific review board, but I'm wondering if there should be more active linkage with the commissioners about what we were looking for as well, or whether your view is that you see enough right now from your point of view. So there's a very long question.
I could pull a few things out of that. The first part, so relative to this slide, I think I remember we talked about what the schedule could be for review. And then I think we had a 3-year cycle of the stock assessment being revised every 3 years. And then trying to match that to when the SRB would be involved and review it, and when it would be ready for an external reviewer. So I don't think the SRB decided on the cycle as much as it was recommended to us, and we just kind of shifted things for the timings to work. That's what I recall from that particular discussion. I'm not sure we recommended an external peer review every 2 years. That sounds a bit too much, a little too frequent, I think, because the whole stock assessment is revised, and there's a kind of a major overhaul potentially every 3 years, and that would be the time I would think an external peer review would be helpful, and I think that's what we tried to line things up for. So how do I think the SRB is working? So one of the things that came out of the external peer review was that they were very positive about the stock assessment and about the science. And that obviously reflects very well on how things are run at the Secretariat and the staff and all the work that they do. That's definitely all about them. There's a little bit about the SRB process kind of promoting that to happen as well. So as I said, we're looking to find errors and problems and have them fixed before they cause further problems down the line. So hopefully you should expect a very positive peer review in these. You shouldn't expect an external reviewer to come in and say, you guys are doing it all wrong. Then I would say the SRB process is not working. It's one of those things where you know when it's not working, but not always when it's working. It seems like, well, do we even need this? I also mentioned somewhere way down there, at the beginning of every meeting, this first one, we have the input from party reps, and that's actually your question. It hits every time we have a meeting and the party reps say, oh, commissioners have these questions. And I think, well, it'd be nice if we could sit down with the commissioners and actually talk to them. And that maybe is one thing where, this particular venue may not be the place to do it, but maybe having maybe an in-camera meeting with the SRB once a year, something like that with the commissioners might be helpful. Does that answer your question?
Yeah, I think maybe I just summarized, I think from your point of view that generally the SRB is working well. It's kind of matured over the years and made some linkages, strong ones, both with government science advisors and as well with the IPHC science staff. And I guess for myself, I would endorse the idea of having some stronger linkage with the commissioners and maybe having an opportunity to meet with the SRB to get a better appreciation of some of your recommendations and why you've arrived at them more than just at this meeting. Mm-hmm. And then I think I noticed in one of your recommendations that in future peer reviews, external ones, that the SRB be included. And personally, I would endorse that as well. I think that's a good idea.
Yeah, that was one of the things I didn't, I thought it was in, maybe it was one of the recommendations I didn't put in here, but I mean, we're all involved in stock assessment. We, you know, as I said, we understand what this review is about. And in choosing a particular external reviewer, it could help for us to give you some ideas about who might be a good external reviewer to use. Sometimes when you go to a general CIE or something, you get somebody that may not be ideal for you. So, and the other issue we talked about was that at certain times in the development cycle, there may be certain issues where you need specific expertise. So for instance, in Hake, every once in a while, they need a more specific review of the acoustic survey. And so that's the expertise that they're looking for at that time.
Other questions? I might have one. I think it was slide 3 or 4. If you could go back to slide 3 or 4, please. This one.
That's a meaty one.
Yeah, and then slide 5. I guess my question is the context of this recommendation, and just to be clear, whether it had any reference or relevance to, for example, the U-26 accounting issue that we've discussed yesterday and that I raised some concerns about yesterday in terms of implementing this year with some of the, in my mind at least, and certainly in a lot of folks that I've talked with, still a lot of questions about how that U-26 accounting works and how those fish flow through the system, and whether there's an underlying assumption that those U-26 would otherwise recruit to O-26, and some hesitation about moving forward with that accounting, notwithstanding the fact that we did last year agree with an expressed intent to do so. There are so many questions that still abound in my mind with that. I'm wondering if this recommendation had any context any relevance in the context of that question.
Well, I think one thing about the in the MSE context is that it's a simulation context, so you can look at a specific issue like trying to think of how you could try to manage, let's say there was a bycatch management approach that was, say, an abundance-based management approach for U-26 fish. And you could put that into the management system and see, okay, if we had a really aggressive U-26 management strategy, how might that play out towards fish available to directed fisheries? And you could kind of dial that knob back and forth to see how sensitive the outcomes are to that. And as I said earlier, accounting for the sensitivity to all these other factors. I mean, there's growth, especially in bycatch management. There's going to be downstream effects because of movement. You know, there's a lot of things that go into what the ultimate outcome is. And it's at this stage, given the current state of knowledge about those interactions, it's probably better as a what if set of scenarios rather than a prescriptive, set of recommendations. The best I can do on that.
Okay, thank you, Dr. Cox.
Okay.
Any other questions on this report? If not, I think we'll go ahead and go on to Dr. Hicks. Next item is a management strategy evaluation update from Alan Hicks.
Okay, good morning, everybody. Glad to be here today. And I arrived last night, but I was able to listen yesterday to the meeting, thanks to the webinar and those who were running it yesterday. I'm going to present to you today a little update on the management strategy evaluation. Every year we have two, what I call MSAB, or Management Strategy Advisory Board meetings. Those meet in May and again in October, and we work through a lot of the details of this, and I'm going to try to summarize all that work in about 20 minutes here, so it's going to be a challenge. First, you may be asking, what is Management Strategy Evaluation, or MSE as I'll call it? And that's basically a process, as Dr. Cox was explaining there, to evaluate harvest strategies, develop a management procedure that's robust to uncertainty and meets defined objectives. So it's really important that we're trying to develop a management procedure that meets defined objectives. And there's many different parts of this process. Defining goals and objectives. We've been working with the MSAB and the commissioners on that, and I'll give you a lot of presentation today on that part. Defining management procedures. What are the management procedures we actually want to test and see if they fit, if they meet our objectives? And I think that begins to answer the question Commissioner Oliver was asking about. They have a procedure in place. Do we know if that's meeting our objectives? The MSE is the right tool to examine that. The Secretariat staff go away, do a lot of simulation evaluation, and then hopefully we apply that and put it into practice, into management. And I'll be talking about all of these boxes today. So what is a harvest strategy, or a harvest strategy policy, or a management procedure? Well, they're all similar things, but I'm going to use the example of our current interim management procedure to try to explain what these are. So yesterday, Dr. Stewart talked a lot about the interim management procedure and the sort of coast-wide part of it and the distribution part of it. The coast-wide part of it's what we're calling scale here. And the management procedure has science inputs as well as management inputs. You can think of a science input, something like the coast-wide assessment. That provides a lot of information, but we also have management inputs such as a reference SPR level, this 46% that Dr. Stewart was talking about, as well as a 30-20 control role. And so if you think about it on a coast-wide level, that scale part of there, we have the coast-wide assessment, these reference fishing intensity coming together to supply with the total mortality. This is how Dr. Stewart gets his total mortality table and all those tables produced. Now the total mortality, we take out of that the U26 non-directed fishery discard mortality, that portion of it, what's remaining is called the TCUI. That TCUI is on a coast-wide level. We care about the regulatory areas, right? And so we want to distribute that TCUI out to the regulatory areas, and we have an interim management procedure to do that, which is composed of stock distribution from the set line survey, defined allocations for 2A and 2B, as well as relative harvest rates. And we'll talk a little bit about all of those. So that's how Dr. Stewart's been creating his decision table and mortality tables. Those now go to the commission in the decision-making process, and that's where the policy's coming in. And they make decisions on the actual catch limits by regulatory area. So that's a really brief understanding process, and when I talk about the current interim management procedure, this is sort of an illustration of that. So the MSAB is working under a program of work. And this program of work has a delivery deadline in 2021 at our next annual meeting. And so this is called a Gantt chart, and you can see a little bit of progress and I pointed to where we're at right now. The key to get from this is that there are things we're still working on and there are things we're delivering at this annual meeting. In particular, I'm going to be describing the goals and objectives that the MSAB has come up with in concert with the commission guidance. And we'll describe those goals and objectives for at this annual meeting, and then some updates on how the MSE process for the remainder of the year before the next annual meeting. I note down on the bottom here is an external peer review, and we're planning to bring in independent external reviewer to review the process of the MSE and the results that are going to come out later this year. So more specifically, I want to identify some of the things we've been doing at the MSAB in 2019 with the 2 MSAB meetings shown in orange and then this annual meeting at the bottom in blue. Basically, we've done a lot of work in the MSAB this year, many different things, but what I'm going to report to you today are sort of the goals and the objectives and the management procedures we've identified to examine in 2020, basically. So in 2020, there's going to be 2 MSAB meetings. One in May this year will be in Courtenay, BC. And I invite anybody to come to that meeting, especially if it's close to you. We try to do a little bit of outreach with this. You can find more details on the IPHC website. We'll be reviewing preliminary results at that meeting and identifying where to go forward before the October meeting, which always occurs in Seattle at the IPHC offices. That October meeting is going to be very important this year. That's where the MSAB will come up with their recommendations. We'll be looking at and reviewing the final results. And then I will be bringing to the annual meeting in January 2021, which I believe is in Victoria, BC, a presentation of that complete MSE. In complete, I mean it encompasses that entire management procedure of scale and distribution components. So I just want to note really quick that we're working as a team here. We have what I call the MSE team with Dr. Steve Barikoff, who's our lead programmer, with a 2-year appointment ending in October, and he's involved with doing a lot of the programming of that simulation analysis part of it. Dr. Piera Carpi was brought on this year, in April this year, with a 2-year appointment ending in April 2021. And she's been helping me a lot with other parts of the programming as well as other analyses and literature review. So there's a team of 3 here, and you'll see us walking around, or at least Dr. Carpi and myself. Feel free to chat with us about anything MSE related or MSAB, how you can get involved, etc. So let's get into the meat of this. And I said I would be presenting a little bit about objectives and what the MSAB came up with objectives for this year. So really there's 4 general categories of objectives. One is keeping the female spawning biomass above a limit. That's a good thing. We need spawning biomass out there, produce more halibut. Objective 2 is maintaining the spawning biomass around a level that optimizes activities. That's good to try to optimize fishing activities. And then, okay, yeah, good. Thanks. Yeah. All right, is this on? Yeah, oh, that's better. It's not as responsive now, feeding back. Okay, and then so a third objective is limiting catch variability. And really what that's about is we don't want the quota to be changing by huge amounts from year to year. We don't want to go from 60 million pounds to 30 million pounds back to 60 million pounds. That just doesn't work for really anybody. And then providing directed fishing yield, make sure that we have the opportunities to go out there and catch halibut. So before I get into the details of those objectives, I want to give you a little bit of background and remind you of a slide that Dr. Stewart presented yesterday on reference points. And I believe this is the exact slide that he presented yesterday, where he defined there's a biological limit of this 20% of spawning biomass, a fishery trigger of 30%, and then there's a concept of a spawning biomass target, which is currently not specified, but the commission asked the MSAB to particularly think about that topic in 2019. We have a reference fishing intensity level of 46%, or that F46%. And then many different management agencies use the concept of a limit F, a fishing intensity that you just shall not exceed. And we really don't have one of those specified in our current management paradigm here at IPHC. So remember this slide, and we'll come back to it after I talk about objectives and management procedures. What helped us in defining objectives and thinking about those reference points is some work that Dr. Carpi and I did on maximum sustainable yield, or MSY related reference points. And really just briefly in a nutshell, we just wanted to determine what are spawning biomass levels that would produce maximum sustainable yield? What are fishing intensities that would produce maximum sustainable yield? And that will help inform what types, where we should be setting our objectives and sort of reference points around that. What we found is that a relative spawning biomass, that's the spawning biomass relative to an unfished spawning biomass, to produce MSY, that's somewhere around 20 to 30%. And then a fishing intensity, or this SPR value we hear about, is more along the lines of 30 to 35%. This isn't groundbreaking news. I mean, there's been many analysis done in the past that show this is really reasonable values, especially for a species like Pacific halibut. What is really interesting about this, and as you know, Pacific halibut are very dynamic. They have a weighted age that changes greatly. You think about halibut used to be really big, now they're just not getting as big at age. And there's also these, we've identified recruitment regimes where there's periods where recruitment seems to be good and then environmental periods where recruitment seems to be bad. We did this analysis looking at those different types of regimes and those dynamics, and we found that these reference points are actually quite stable across those different regimes. So they would be really well suited to define a long-term objective that is stable across different regimes and the dynamics of Pacific halibut. So that was a really useful result in moving forward. So here's some objectives, and just to orient you to these tables, we have on the left sort of a description of the objective. We have in the middle there, and then we have what's called a time frame and a tolerance on the right, and that tolerance really helps us define the risk we're willing to accept in not meeting that objective. So for example, this objective is that primary biological objective of maintaining the female spawning biomass above some lower limit, and that limit's defined as 20% of unfished spawning biomass. So basically think about it like this. We don't want the spawning biomass to get so low that there might be detrimental outcomes. Say they recruitment is impaired or something like that. And we've identified 20% as a potential threshold for that. For a number of reasons, mainly because that's what's been used in many other fisheries and research that's been done historically. So we have that threshold on a coast-wide level. We just want some spawners out there, but we also want there to be spawning biomass in the different biological regions throughout the coast. As Dr. Planas showed earlier, spawning occurring in certain areas might lead to larvae in other areas. And so we don't know where in given environmental conditions, what spawning area is going to produce the most larvae. So we just want to maintain spawning biomass across the entire range of halibut. And we've identified that objective to look at that. Second objective here was asked by the commission for us to consider, and this is a target spawning biomass. What would be a good spawning biomass we just sort of want to be around most of the time? And using that analysis that I described a few slides ago on the MSY-based reference points, we came up with, well, a relative spawning biomass of about 30% is a precautionary MSY proxy. So maybe 30% would be a good target. But MSY, and there's been a lot of research on this, managing to MSY has some risks associated with it, and also managing to MSY may not be the most optimal solution for economics. It might not maximize the economic yield. So in discussions with the MSAB, we came up with the number of a relative spawning biomass of 36%, has a buffer to avoid exceeding MSY and taking on that extra risk, as well as it may result in better optimal economics in the fishery as well. So we've identified a target biomass of 36%, and being a target, that means you want to be above it 50% of the time and below it 50% of the time. So those are two spawning biomass related objectives. Fisheries, a real specific fisheries objective is the stability objective. And that's really annual changes in that coastwide TCY or by regulatory areas as well. And so these different objectives try to get at that. And what the MSAB has really identified is they do not to have changes that are really greater than 15% a lot of the time. They don't want to see changes in the quota more than 15%. Maybe once in a great while, but let's try to keep it below that level. And so we have a number of objectives trying to get at that concept here, where annual change is just a measure of how the quota is changing from year to year. And then finally, the yield objectives. And this is really important. We want yield out there. We want that opportunity to take yield because that's what we do. We fish for halibut. And so on a coastwide level, we want to look at what the yield would be under these different management procedures, as well as on an area level, looking at it within an area specifically, and that's this median TCUI, as well as sort of the sharing across areas, and that would be this percentage TCUI. We've defined a number of objectives to look at that with testing these management procedures. So you're asking, great, we have these objectives. What does that really mean to us? Well, the purpose here, remember, is to evaluate management procedures and see that they meet those objectives. So those objectives are what they're going to do is Dr. Carpy and I will go away, look at those objectives and develop performance metrics that really indicates whether we're meeting the objective or not. And given those performance metrics, which I'll show you in a moment here, we can look at, did we actually meet those tolerance levels for those biological objectives, for example? And having that tolerance defined is really good because, as Dr. Cox explained, you want to sort of narrow down the suite of management procedures you're looking at. We might look at 200, 500, 1,000 different management procedures. Well, we're not just going to just spit them all out to you here to decide which one you like best. These objectives help us narrow those down, and that tolerance really limits the scope that we would examine. Once we have that smaller set of management procedures that we feel is working, well enough, then we'll begin to evaluate trade-offs, and that's the trade-offs, stability and yield sort of things. And so we'll look at the trade-offs between stability and yield. The harder you fish and the more yield you take, typically the more change from year to year your quotas will be. So there's a real trade-off there that the MSAB will evaluate. And then I note there'll be a lot more performance metrics and statistics considered that are associated with other sort of minor objectives identified by the MSAB and the commission over time. So using the coastwide MSE that we've presented, basically I think about the last year and a half, using the results from that coastwide MSE, I want to apply this current set of objectives and see where we're at on that scale portion of that management procedure. Here we have the opportunity to actually evaluate the SPR, the fishing intensity and control rule, and a constraint potentially, and see how it meets the objectives. So what I'm showing here on the left is the probability that the relative spawning biomass falls below 20%. So we want that probability low. So really what we don't want to do is be in the red area of this plot. That means the probability is high that we go to a really low level of spawning biomass. Not good. And then what each of those lines and symbols represents is a different management procedure that we tested. For example, the one that's obvious there on the left is the black diamond, and that's no control rule. And that means we don't start decreasing fishing intensity when the spawning biomass gets low, and we just keep fishing at that fishing intensity, which might work for some fisheries. But apparently here there's a high risk when you're fishing at higher fishing intensities. And so I should note on the horizontal or x-axis there, that procedural SPR, from left to right is increasing the fishing intensity. You're fishing harder on the right side of that plot. Thus, you're fishing harder. There's more chance of small biomass. And so these other points and lines are different management procedures, such as different control rules and different constraints on the annual change in the TCUI. So if this said, oh boy, you need to reduce the TCUI by 30%, a max change both 15 means we're not going to change it by 30%, we're only going to change it by 15%. And that's either up or down. So really the key thing there is on the left, the probability of falling to really low biomasses is not a problem for most of these management procedures. It is for a couple, but really above a procedural SPR of 40%, we're pretty much meeting that objective. So that's good to know. On the right side is this objective of meeting the target 36% relative spawning biomass. And what you can see there is that many management procedures are sort of clustered around that middle, right where that red and white divide is. So we really don't want to be in the red, but you can see most of the management procedures fishing at a fishing intensity less than 40%, or the SPR is greater than 40%. It's a little backwards. That you're in the white area and you're meeting that target objective. So really what this is defining, and I'll click to the next slide here, is that across, again, the horizontal axis are these fishing intensities. When you get when you're fishing harder than the 0.40, you're not meeting that target objective anymore. And that is why I've sort of made it a lighter color in these plots, because those are really management procedures that we shouldn't be considering anymore. We've narrowed it down in that context. So really, we've focused in now on the left portion of all of these plots, and now we can look at yield on the top there, and we see the harder we fish, the more yield we get. But we can also look at it a different way, and the probability that the yield might be less than 34 million pounds in a year coast-wide. I'm sure everybody can agree that's probably not where we want to be very often. And you can see that that probability is at its lowest point around a 0.42 or 44. So that's a really interesting result that we found from these MSE results, is that you typically keep the total mortality coast-wide above 34 million pounds with a SPR around 43%. On the bottom is stability in the yield, and that's how much it changes from year to year. On the left, you just see sort of that chance or the probability that the quota will change by more than 15% in at least 3 years out of 10. And you see that any management procedure along the top there are ones without a constraint, and you see that probability is really high. There's just natural variability in this whole system of the dynamics, as well as the uncertainty in the assessment, that results in quota changes that can be high from year to year. But introducing a constraint really limits that, as it would, because that's an engineering way to limit the change from year to year. And then on the bottom right is, again, a different way to look at that, just the average annual variability from year to year. So what did we really learn from this? Well, we've identified really a small range of management procedures that might work for the IPHC. And now we can maybe update this reference point slide that I showed earlier that Dr. Stewart presented yesterday, indicating the interim management procedure. So this is considering now with these results, should we be looking at what our current interim management procedure, how can that be changed? Well, we have on the top still the SB20, the SB30, part of our control rule, as well as a biological limit. Red colors here are related to objectives. Blue are related to the management procedure. So you can think of the 30-20 is the control rule management procedure, and that's where those reference points come from, and that's why they're blue there. The biological target's an objective, and that's now, we've defined that as SB 36%. One change, which you might have noticed on here, is that the MSE results are supporting a reference level fishing intensity of F43%. On Dr. Stewart's slide he presented yesterday, that said 46%. So these results are indicating we might be able to, that we can meet our objectives by fishing at a F43%. And there's still not a defined F limit, but the MSAB has been thinking about that. So this is just a way to look at that control rule, all those different reference points on there. You see how the control rule, the fishing intensities on the vertical axis, the control rule, when the stock status gets low, it just basically reduces the fishing intensity. And what we found is a control rule is a really important part of this management procedure as well. It really limits the probability of getting to small biomasses. So that's the objectives and the results from the coastwide MSE. Another big part of the MSAB this year was identifying management procedures to investigate. Here's scale portion management procedures, the coastwide level, that fishing intensity SPR, the control rule, and various constraints, which we've already investigated on a coastwide level. Now we want to combine that with distribution procedures. And so just thinking about how distribution works, we're going from coast-wide and we want to end up at some regulatory area allocation. We may pass through regional on the way to get there. We're investigating ways to do that, and I'm going to be testing some ways to do that. And then that end result is the annual regulatory area adjustment. That's the policy part of this. And I just want to note, all that's really required is we're starting at coast-wide and we want to get to regulatory area. So going through region is an option, and we're seeing if that helps meet our objectives. I want to pause for a moment because one important part of the current interim management procedure are these relative harvest rates by region. So if you remember from Dr. Stewart's presentation, he talked about a relative harvest rate of 1 in eastern areas and a relative harvest rate of 0.75 in western areas like 4A, 4C, and E, 4B, and 3B. What we looked at with the MSE team is we wanted to see what were the considerations that led to that, those current relative harvest rates. And we looked in the past, and there's a summary in the documents if you want to read about it, but there were a lot of considerations that go into the definition of these relative harvest rates. And a really important one is movement, immigration and emigration. If there are areas that are what we call sinks, rather than sources, that means fish are moving into those areas and tend to congregate in those areas, and they might be able to sustain higher harvest rates. And we have an understanding that adult fish generally move from east or west to east for Pacific halibut. There were other considerations, trend in the data, uncertainty and observations, et cetera. But one part of this determination of relative harvest rate was the productivity by the different areas. And what we did is we looked at a productivity analysis by region. And we did this through a yield per recruit analysis in 3 different time eras. And we picked these 3 time eras because 1985 was a different selectivity. The fishery was selecting fish differently, and fish tended to be really large back then. 1999, the productivity or the weighted age and the selectivity changed. And then 2018, as we all know now, fish tend not to get as large at age, and selectivity has changed for a number of different reasons. But what we found is that in these are relative to all to region 3, that region 2 and region 3 have similar productivity on a sense of how should you harvest it to maximize the yield per recruit, yield per fish, you can think about that as. But region 4 has actually changed. It seems to have a change in productivity in years, suggesting that in more recent years, its relative harvest rate is more similar to areas 2 and 3 than it was, say, in 1985. So we found that as an interesting result, and that there is this dynamics of changing relative productivity. But remembering this productivity analysis is one part of all of this. And as we talked about with bycatch or non-directed fishing mortality and other things, the MSE is really the appropriate tool to ask those what if questions. And that's, I think, the point Dr. Cox was trying to make. So what are we looking at this year in the MSAB? We're going to look at a lot of different elements to distribute the TCY from coastwide to region. These are things like going from the coastwide directly to regulatory areas or region, the relative harvest rates. We're going to be looking at some options there. The defined catch limit for 2A and a defined percentage for 2B. We're going to look at different options for there. So really, this is where we get to experiment, come up with ideas, and see how good those ideas might work in practice. So at MSAB 15, the meeting in Courtenay, BC, I believe that's May 11th to 14th, but check the website to make sure. You're all welcome to come. We'll be presenting results on this. We'll come out of that MSAB meeting with a whole bunch of recommendations, I'm sure, confident of, and we'll have a lot more work to do over summer, and then we'll come back at MSAB 16 with results on management procedures that incorporate scale and distribution for presentation at annual meeting 097. So I just want to point out, we got a lot of work ahead of us. There's many different combinations of these. We'll have hundreds and hundreds of management procedures, and we're going to work our hardest to bring those down into a really informative set. And then I just want to finish off with some recommendations. basically noting the paper that's online. That's a really informative paper that goes into a lot more background than I've presented. We have 10 primary objectives and metrics, including the target spawning biomass objective that we would like the commission to consider. We've found in the coast-wide results that the SPR greater than 40% with a 30-20 control rule and some type of constraint meets all of the objectives we've defined. And we're really recommending, the MSAB came forward with a recommendation of an SPR 43% to update the interim management procedure. The objectives and the results really support that. And then a bunch of notes that we're going to be investigating scale and distribution in the future for delivery at the AM097. And with that, I'll take any questions. Thank you.
Mr. DeGree.
Thanks for the presentation, Dr. Hicks. I just relating back to what we heard from Dr. Sean Cox and Chris's question earlier, the discussion of bycatch. How has MSAB dealt with the bycatch in the presently, and what was the rationale for dealing with it in that way?
Yes, thanks, Commissioner DeGree. That's a really good question and something we did consider a lot. So I joined IPHC in April 2016, and I jumped right into an MSAB meeting. And really what I found there, I think there might have been 10 people at that MSAB meeting. It seemed like some people were starting to lose interest and didn't think this was going anywhere. So my real goal in the beginning was, let's start getting results out. We've done that in the last couple of years. To do that, we had to start really identifying, prioritize different tasks. So we had a long discussion with the MSAB. What should we be prioritizing? Should we look at scale first and then distribution components, really focus in on the directed fishery and what we should do with the directed fishery? And the agreement was yes, let's focus on the directed fishery. Let's get stuff in front of the commission and start making decisions. To do that, we realized that bycatch is a whole nother topic, a big issue. And we decided to put that into a category that we call scenarios. Something that we're not looking specifically as part of the management procedure right now, but more as part of the process that's occurring and has some variability around it. So given that advice from the MSAB and the commission at that time, okay, let's go forward focusing on directed fishery, treat bycatch as a scenario, something that we're not looking to control right now. We can in the future, but we made the decision in the simulations to basically simulate it as a certain amount of bycatch occurring every year. On average, I think in the simulations it's 7 million pounds, but it has variability. Some years it might be 10 million, other years it might be 4 million. So really that fits into the key is we're trying to identify management procedures that are robust to uncertainty. We're just putting bycatch into that uncertainty category. In the future, we can pull bycatch out, put it into the management procedure category, and actually identify different ways that bycatch might be incorporated into the management procedure, and more specifically, and what the results are and how that will meet our objectives.
Thank you.
I guess I'll follow up with a similar question on maybe slide 23, Dr. Hicks. 23.
23. Okay.
You make reference obviously to the 2A catch limit and the defined percentage for 2B. And, one of the other things that came out of last year's meetings, I'll go back to my U26 discussion, was in essence a policy decision to look at full accountability for U26 and an expressed intent relative to doing that in 2020. But as over the intervening time period, I've come to believe it may have not been a fully informed policy decision. And I'm wondering if you have this, I don't know if this is a fair question or not, if you have any thoughts on that in terms of this MSE process and how that MSE process could better inform how we deal with that.
Yeah, thanks, Commissioner Oliver. that is something that is on a lot of people's minds right now. Incorporating the decisions the commission made related to 2A and 2B last year was really simple for the MSAB to do. And it just fit nicely into the program of work that we had already identified. The concept of the U26 bycatch, or the non-directed fishing mortality, is something that is a little bit more difficult for us to just quickly put into this process that we're working on right now. So about 2 years ago, we defined this program of work, and we've been really down that path with the option that bycatch is really being treated as the scenario for right now. Bringing bycatch in would really widen the path, or actually cause us to divert off of the current path. And so the MSABs decided, let's get these results out. We've got just less than 1 more year to do this, which is a little scary to me, but motivating. And then let's focus again on those non-directed fishing mortality issues potentially in the future. So something the MSAB does need to talk about, and the commission may want to consider as well, is where's the MSE going to go after 2021? We do have a 5-year program of work, but it's not well defined after 2021. And there's a lot of things that we could pick up and investigate in the MSE. But it really is the appropriate tool for the U26, because it's going to have that feedback of not taking U26 or non-directed fishing mortality in a certain area. What is the actual population dynamics and feedback on the other areas, instead of what the analysis Ian has done is a really good look at currently what would that look like, but the MSE is going to have the whole migration feedback in that as well.
So just to make sure I heard you right, you said it is or is not the appropriate tool to look at that?
The MSE is the appropriate tool to look at the U26 non-directed fishing mortality, all the non-directed fishing mortality in general, because it's going to give you the feedback of migration and movement of not removing fish from certain areas and how those do affect other areas.
Thank you, Dr. Hicks. Look, other questions for Alan? Okay, if not, thinking it might be time for a morning break. And so let's take a break till 11:10. 11:10, thank you. Then come to order. Thank you. I think we're now moving on to item 10.2, which is the strategy, Management Strategy Advisory Board reports from the MSAB co-chairs. And I'll turn it over to Carrie McGilliard and Adam.
Thank you. We are the co-chairs of the Management Strategy Advisory Board, which is the board that Alan was just talking about in his talk. I am Carrie McGilliard, and this is Adam Kaiser. And we're here just to give you a recap of the past 2 meetings of the Management Strategy Advisory Board. One was in May in Sitka, Alaska, and the second was in October in Seattle, Washington. And we got a lot done this year. We did what we did in 2019 was we finished defining objectives for distribution and scale, and so that's defining objectives for how we distribute the catch limits among areas, and also what the coast-wide fishing intensity is. We reviewed multi-area model development. Alan, as he just described, has a model that models all of the biological regions and then has a way to get at what might go on in particular regulatory areas. And we identified management procedures for distribution and scale. And so that will allow us to move forward to have some alternatives to analyze about how different options for how catch limits would be distributed. You can see the objectives for distribution and scale in the MSAB reports. So the MSAB report from meeting 14, recommendation 1 and recommendation 2, if you want to look in detail, lists the objectives for both distribution and scale. And we also identified management procedures for distribution and scale, which are in the same report, and you can see in recommendations 3, 4, and 5. We talked a lot about primary objectives, and I think we talked about this enough to put it to bed for a little while. We have 4. I can't see the screen because I'm too short. We have some general objectives. The first one that gets prioritized is keeping female spawning biomass above a limit to avoid critical stock sizes and to conserve spatial population structure. That's a general objective, and then more specifically, we have measurable objectives, 2 measurable objectives that go along with that we've defined at these meetings. One is to maintain female spawning stock biomass above a limit reference point at least 95% of the time, and the other is to maintain a defined minimum proportion of female spawning biomass in each biological region. So that was a new development that we added this year as we were talking more about distribution. General objective 2 is to maintain spawning biomass around a level that optimizes fishing activities. This is also a general objective that we really worked hard on developing this past year on instructions from the commissioners. And we developed a measurable objective to maintain the coastwide female spawning biomass above a target reference point at least 50% of the time. We also worked on a general objective to limit catch variability, and we've had this objective for quite a while now, and that means limit annual changes in the coastwide TCEY and also to limit annual changes in the regulatory area TCEY, as that is what would affect people the most. General objective 2.3 is to provide directed fishing yield, and you'll see that we have a number of objectives under that. They all get at something that's just, you know, a little bit of a different aspect of provided directed fishing yield. So one is to optimize average coastwide TCEY. One is to optimize TCEY among the regulatory areas. One is to optimize the percentage of the coastwide TCEY among the regulatory areas. And then we have one that is to maintain a minimum TCEY for each regulatory area and to maintain a percentage of the coastwide TCEY for each regulatory area. So as Alan goes through and he does the analysis, he is going to be evaluating whether each of the management procedures on the table can meet each of these objectives. You can also read more about this. There's a complete table in Appendix 5 of the MSAB 14 report. And there is the link right there if you wanted to write the the link down, or I think that it's just provided in the general with all the rest of the documents from the meeting. And I'll hand it over to Adam now.
All right, so we, Dr. McGillier just went through recommendations 1 and 2, which were focused on the objectives. That's something that's taken up a fair bit of time of the MSAB's, or a fair bit of the MSAB's time over the past couple of years. Recommendations 3 through 5 are focused on distribution and on fishing intensity. This has all largely been covered by the presentation we just saw from Dr. Hicks. We're taking this as an opportunity to present the MSAB's perspective on it. So the first recommendation, recommendation 1 here, oh sorry, I mixed the numbers up. Recommendation 1 is the target. Recommendation 1 is regarding the biomass target, which was in response to a commissioner request from late 2018. The biomass target identifies the I guess, desirable biomass over the long term. And while I think many here are familiar with the concept of maximum sustainable yield as a biomass target, Dr. Hicks, just before the break, outlined a few, I guess, challenges, considerations that decision makers, stakeholders should be aware of when identifying MSY as a potential biomass target. Perhaps potential precautionary proxies to address some of the uncertainties with MSY as a biomass target. And so it was with these considerations that the MSAB recommended a biomass target objective that was to maintain spawning biomass above a target reference point of 36% of the relative spawning biomass over 50% of the time. So this is a level that's slightly larger than the fishery trigger, which is the top end of the ramp in the harvest control rule. We'll have an image in a few slides here to make it a bit clearer. The next recommendation is recommendation 3 we want to draw folks' attention to, and this is specifically regarding a target fishing intensity. Now this recommendation is consistent with past iterations that folks have seen from the MSAB over the past couple of years. Just benefits from some of the refinement of the simulation work that the Secretariat's conducted in 2019 in particular. So the specifics of this fishing intensity, this scale, is that the MSAB recommended a fishing intensity of SPR 43% with a specific harvest control rule. Here it's called 30-20 harvest control rule. And then some additional constraints, some smoothing mechanisms to help reduce variability in annual catches. The MSAB, though, also noted that that this scale recommendation is only one component of the management procedure. There is still work underway on distribution. So that given that we don't have all of the components of the entire management procedure, there's still some more work to be done on distribution. We also identified a range of SPRs to be considered within subsequent procedures that will be evaluated over the coming year through 2020. So while the target fishing intensity recommendation is SPR 43%, we should also consider additional procedures that are within this range of 40 to 46. I'm just going to skip ahead one slide. So this is the harvest control rule. When we talk about 30-20, 20 is the limit, trigger is the 30%, in target in the red is the recommendation 01 that we just summarized. So this is the scale component of, or the harvest control rule that is being evaluated, or that we've identified some components for that's being evaluated over the coming year. Okay, so very quickly, we've gone through a couple of the recommendations the MSAB has put forward with respect to scale. The next couple of recommendations are more specifically around distribution of catch opportunities. So the MSAB had identified a very, very long list for Alan and the MSE team to consider as management procedure components for distribution. We talked about different operational control points. That's the inflection points of this harvest control rule here. We've talked about different reference fishing intensities, SPR 43, 46, 40. We've talked about different ways to limit catch variability between years. And we also talked about relative harvest rates, which Dr. Hicks had just presented before the break as well. A number of smoothing indices, or smoothers to indices, were also considered. as well as different ways to distribute harvest opportunities amongst regulatory areas and amongst biological regions. The challenge, of course, is trying to figure out how we select those suite of management procedure components into a workable set of procedures for the Secretariat to evaluate. So in our last meeting in Seattle, the MSAB identified 17 candidate procedures for the first round of evaluation. These are procedures that would consider both scale and distribution. We then, a little begrudgingly, prioritized 10 of them. We really want all of them, but we know there's only so much work that can be done. We were already asking a lot of the Secretariat. We prioritized 10 of these procedures to be evaluated and presented at the meeting coming up in Courtenay, British Columbia. We did document all of our suggestions, though. As noted on the slide here, there's a complete table of these candidate procedures in Appendix 6 of our last report. So, given those recommendations, where to from here? What does it look like for 2020? As you've already heard a little bit earlier, in May, we're going to be reviewing the first cut of the recommendation, or first cut of the evaluations and simulations that the MSE team has been working on since October 2019. It's an opportunity for the MSAB to refine those recommendations, to figure out where maybe we need a little more exploration, or maybe we didn't quite characterize one of our requests as clearly as we could have. That work would then be reevaluated or continued through summer 2020, where we get to our almost pinnacle MSAB meeting in October 2020, where we will review these results to rank management procedures, identify the trade-offs between our different procedures, and ultimately provide a recommendation for the commissioners at annual meeting 97. So that's the very brief summary of the MSAB's work over the past year. Before we turn over to questions, though, I just wanted to acknowledge a number of the MSAB members that are in the room. These are long meetings, 2-week-long meetings every year, and there's a number of folks here that have put in an incredible amount of time over the past few years. It's not an easy task. It's a big ask of folks. So at least as co-chairs, we'd really like to express our appreciation for the work that those members have put in. Okay.
Questions for the co-chairs? Seeing none, thank you very much. I think we have a public comment break here. Items 7 through 10.
Thank you, Mr. Chairman. So as we did yesterday afternoon, as I call your name, we have a microphone at the front table here. If you can come up here and make your comment. I'm going to call people first for the topics that we've just heard this morning, and starting with Eddie Ferguson. And following him will be Bernie Burkholder.
Morning. We're trying to keep the biomass up. I think everybody agrees that's a good thing. I just don't think that the bycatch has been looked at enough. It's being looked at, but too much bycatch is going to make the biomass just fall right on its face. That's all I've got this morning.
Thank you. Next up is then Bernie Burkholder, followed by Brian Ritchie.
My name's Bernie Burkholder, and I'm here today representing the Sable Fish and Halibut Pot Association. And really, I don't know if the question is appropriate, but you know, I would like to look at mortality discards. I think it would be prudent to look at that for pots as well as hook and line, and how that would compare to careful release and cutting of the ganglion, and also some blood chemistry. So I don't know, has any of that been done, or are you considering that, or is that something that maybe is, you know, don't mean to put you on the spot, but it just seems like, you know, if we're trying to get these fish back live, it's probably looked good to look at something out of the box a little bit.
And thank you. We will note that. And I know the scientific team will talk about that in context of the discard mortality studies that they're doing. And we'll also bring that up to the Research Advisory Board. It meets in a couple of weeks because that's one of their continuing topics is discard mortality.
Thank you very much.
Next to the microphone, please, is Brian Ritchie, followed by Forrest Braden.
Good morning. My name is Brian Ritchie. I'm a stakeholder from 3A and Homer. Just given what we sat through this morning and presentations by the Management Strategy Advisory Board, as well as Dr. Planas, I guess I had some questions in regard to the current distributions of the stock and current distributions of TCEY. It looks like we can assume that there's a fairly high degree of emigration of fish and larvae from areas that are in the south and east of the IPHC's management domain. My interpretation of this and the basic oceanographic effects on the stock distribution could be misguided, but it seems to indicate that these areas essentially are net exporters of fish to the rest of the Gulf of Alaska. Noting that, what the MSAB has just shown us, what role is this science going to play in management's current and future distribution procedures for TCEY? And what role does the management strategy evaluation have in the current distribution procedures as they relate to science? I guess, I don't know if that's a question or a statement. I just hope that it's something you guys consider in future decisions for guys like me.
Have them talk about.
Oh, okay, Dr. Hicks may have a response.
Yeah.
All right, thanks for that. That's a really good comment, and I'll just try to answer it from the MSC point of view. Is in the management strategy evaluation in the MSAB, we're considering the population dynamics of halibut, and all the work that Dr. Planas is doing feeds directly into how we're building our models and our understanding of the dynamics of Pacific halibut. So when you're saying how they move and the larvae and the spawning biomasses and how that's all affecting, that'll be part of our model. And that's exactly what we're trying to test for. What is a management procedure scientifically that actually accounts for that type of movement and would work best and meet the objectives of all the different fishermen? So the issue there is, you know, we have some data indicating that, and we have some understandings of oceanographic stuff, but the environment's variable and we have uncertainty in our understanding. So we're going to have to build in a lot of variability and uncertainty into the process and develop management procedures that are robust to that level of uncertainty. Does that help?
Yeah, I think so. Well, maybe I'll catch up with you more after this.
Find me wherever.
All right. Thank you very much.
Thank you. Who's next, Steve?
Next up here is Forrest Braden, followed by Linda Bankin.
Good
Good morning, Mr. Chair, and thanks for this opportunity. This is Forrest Brain for the record, Southeast Alaska Guides Organization. The topic I wanted to just speak on briefly is the discard mortality study that was brought up in Dr. Palanis's presentation. And I just got an email back from the team, and so I see that there's, I think it was 43 pop-up tags slated for the study, but you're trying to secure more. And then wire tagging that's going on. And I noticed in one of the presentations that there were some 10,700 and something odd wire tags done. I don't know over what time period, but there was only 12 recovered. And that ratio was, you know, a little confusing. You'd think that you'd see more of those back. But my main point is this will have a pretty significant bearing on how you determine allocations for the charter industry. So I'm really in support that you're trying to secure more funding for more pop-up tags. I'd hate for this study to be underfunded and then us to, I know that it's better than the age-old formula that we are using probably, but I'd hate for lack of sampling size to occur in something that would have long-term effects on the charter industry moving forward. And that's my comment. Thank you.
Thank you.
Thank you. Next is Linda Benkin, followed by John Moline.
Good morning, Commissioners. Linda Benkin with Alaska Longline Fishermen's Association. I wanted to just briefly talk about the research project that we're working on with the IPHC and then offer a few more general comments on policy for this interim period. Dr. Palanis did mention that we're working on a towed array research project with the IPHC. For the benefit of some of the stakeholders here, that effort is to help fishermen know where sperm whales are in real time and to automate the delivery of that information to vessels fishing in the area so they can more actively avoid whale depredation while they're out there fishing. We'll be putting together a network of fishermen this spring to share that information with as it comes into us from the towed array. And would love to involve as many fishermen, at least this year in the Eastern Gulf as we can, in 2C Eastern Gulf. So if you're interested in that, talk to me. If you can't find me, find Dan, he's taller, and he can also sign you up. I did want to offer a few statements, if I could, on general policy during this interim period. My first would be that during this time of declining abundance, we think it's really important that both of the contracting parties conserve the resource, i.e. that nobody takes a bigger TCUY between the two parties than last year. We need to reduce fishing intensity on the stock until we see stronger recruitment. We'd also like to see you revisit the agreement from last year and the general apportionment to Canada. We never understood the rationale. We think there's some pieces of that that ought to be revisited. We oppose at this point the incorporation of the U-26 compensation. I would have to say first, representing 2C stakeholders where the bycatch is the absolute lowest in the fishery, it's pretty hard to think about compensating areas with higher bycatch. But probably more relevant to the question in front of you is this time, based on the presentation you had yesterday from Dr. Stewart and also from Dr. Hicks today, it's quite clear that the U-26 issue relative to bycatch is very dynamic. There's a lot of factors involved, and it seems to us that it needs to have a good look, as Dr. Hicks suggested, by the MSE process and by the MSAB, that it's not appropriate to roll that in as an interim measure. That needs to be part of a more long-term strategy for the Halibut Commission. And finally, we would urge you at this meeting to identify a fishing intensity that's within the range of what's been identified by the MSAB. I know they're still working on it, but they've given you some guidance at this point that's more conservative than the realized fishing intensity from last year. but within that range. And we believe that once that fishing intensity is committed to by both contracting parties, that there needs to be domestic decisions made by the two parties with full input from the stakeholders, Conference Board and PAG that are part of those countries. And I would stop there. Thank you.
A question, Linda, on the fishing intensity point, because we thought we were doing F46, but it ended up being at F40, realized, I believe, if we were to choose something more conservative than the F40, but more aggressive than the F46, but recognizing it's dynamic and we have to look back at what happened when we get next year, you weren't suggesting we pick a intensity that we would be binding ourselves to during this interim period, but rather for this year alone?
Yes, through the chair, thank you for that question. Yes, that for this year, I think at this point, there's so much flux and change in the resource that this may be something you need to revisit. You have some bookends from the MSAB, and just look for you to stay within those bookends and be more conservative than the realized harvest rate from last year.
Thank you. I don't see any further questions.
Thank you.
The next up is John Moline, followed by Levi Rangel.
Hi, I'm John Moline. I own a fishing charter business out of Seward. I've been running it since 2003. Been through a whole lot of the changes with the CHPs, allocation loss, reallocation. I'm not normally known for being using a lot of scientific terms and everything. I generally tend to ruffle a few feathers by speaking the truth the way it is. The last time I was moved to speak, it was at a NPFMC meeting. It was about reallocation. Nobody really liked the phrase that I used. Reallocation is nothing more than theft. When I reallocated part of my brother's candy bar to my mouth instead of his, my mother took it out on me. That's what I mean by ruffling feathers. In reality, last year I was absolutely flabbergasted when I found out that Area 3A was finally under, but yet somebody told 1 million pounds of fish to pack their bags and move south. That has nothing to do with science and biology. You can't tell fish to move south. It doesn't work that way. To me, I took that more as activism. Personally, I've always respected this board for science and biology, and that really hit me hard personally, that I've been trying everything I can for 15 years with my customers to try to maintain a resource, and then got slapped in the face like that. That hurt me, my business. And now we're talking about if that 25% wouldn't have been shifted south, we wouldn't have been over. But yet now we're talking about big hits in the same area. Why? It doesn't make any sense biologically. None. I would say the IPHC, in my opinion, has had a long-term effect of saying, okay, let's not make a drastic change right now today. Let's look at this because something's obviously out of sorts. Let's take an average, let's look it out, let's look it over. I don't know what you're talking about as far as a 46 and a 36, all that other stuff. But maintaining something that historically we've had for the past 3 to 5 years, maintaining something in that avenue, leveling it out, smoothing it out, that seems to be about the best policy at this time frame. I didn't mean to ruffle any feathers, but I just needed to say it in a way where people would understand where I'm coming from. So I don't really have a question for anybody. Does anybody have one for me?
Any questions from commissioners? Thank you.
Thank you.
Next, please, is Levi Rangel, followed by Kirill Basargin.
My name is Levi Rangel. I'm a charter owner and operator down in Seward. I am, to my knowledge, the youngest owner operator in the business here in Alaska. And when I started my business, there was a whole lot of people that told me you can't do it for a lot of reasons. I see what they're talking about. Since I've been in business, regulations have changed every year. And they don't change at a time where a business owner has time to plan out their business for the year. We're about to head into our season, and we still don't know what the regulations are going to be. That makes it very tough. And now I see why my predecessors are falling by the day. I would urge everybody here to heavily think about what you're doing to the small business owner. It's not getting easier. The best thing we could do is look at long-term regulations. Short-term reaction, in my opinion, is not the best way to regulate a fishery. That's all I have to say. Thank you.
Thank you.
Thank you. Next is Kirill Basargin, followed by Jeff Kaufman.
Hello, good afternoon. I have a comment on a concern from our board. There has been a lot of talk about bycatch, but it hasn't been addressed intensively. We would like to see the IPHC and the management to address and put more percentage and work into the mortality of each species per sector. As of now, we don't see which bycatch or where and by whom it's getting caught by. All we see is bycatch on the reports. We see it as bycatch is a very vital fishery where it's getting caught and tossed, and mortality is in effect dropping the fishery. So if we put percentages into there and who is it caught by, then we can avoid getting the bycatch, and it can implement and put more effort into fishermen having better sustainable fishery. Also, I have another comment that a gentleman here was talking about fishery being allocated from one district to another of the halibut. Say we're fishing 4A and 2C or whomever each district is fishing by, which wherever the depletion is, that's where the drops should be allocated to, but not taken from other districts. It will ruin our fishery, and it will destroy our fishing hood to our future generation. Let's look into the 20, 30 years, but not what we can do right now. That's all I have.
Any questions? Thank you.
Jeff Kaufman next, followed by Chris Sporer.
Good morning. My name is Jeff Kaufman. I'm from St. Paul Island and a 4C D&E fisherman. So I have two comments today and one question for the commissioner, for the staff. My first comment would be that, as mentioned by staff in the presentations, that the handling of the U-26 proportion of PSC or bycatch be part of the agreement with Canada and Washington. And we are urging from our area that we delay implementation or not take implementation in the future on this until it is better understood. And I think the staff pointed out very clearly that this is premature, that there may be implications that we're not considering. Migration is huge, and we're in our area, we're already facing some, potentially a 67% reduction in our allocation. And if we don't include Canada and Washington in the calculations of the U26 proportion of the PSC, it will do further damage to our area. And so we ask that you continue to have the MSAB work on that. The second comment is that we would ask that the MSAB continue to put a lot of effort into looking at the harvest rates and what the implications may be of those. As I mentioned to the U.S. commissioners last night in the U.S. delegation meeting, right now, the way it's currently structured is we're getting the leftovers from the bycatch in that area. And so it's having a real negative effect on our allocations. And then basically with the harvest rate that has been implemented by the IPHC for reasons that are no longer applicable, we're taking 25% of the fish from the Bering Sea and literally putting it in the eastern areas. I think it's premature maybe to do something with the harvest rate at this particular meeting, but I would urge the commission to continue to put or continue to prioritize through the MSAB looking at these harvest rates and making adjustments when we feel like that's fit. And then the question that I have is about the, obviously in the North Pacific Council, we're working on abundance-based management of the halibut resource. And in that process, I've heard many, many times that that the harvest or that the control rule policy of the halibut commission may or may not be implemented if we drop below the B30 or the B20 line. And so there's a lot of doubt in that forum as to whether this is really a valid policy or not, and it's having implications on how we deal with the ABM action, and it's creating a lot of uncertainty there. And so if the IPHC has a policy, a control rule policy that will ramp down the harvest rate at B30 and then shut down the fishery at B20, I think it would be really helpful if this body could forward that policy to the North Pacific Council, understanding that I guess it could change, but anything can change. But what is the current policy right now? It would be nice for the public to understand what that is, that it's real, that there is a policy, and that they should trust in that when working on regulations for abundance-based management of halibut. And so I'm just wondering, is there a policy, and is it something that could be sent to the North Pacific Council?
I think we may have staff that they're in a position to provide a response, Jeff. Yeah, Dr. Stewart's coming up.
Thank you, Mr. Chairman. Yes, the current interim management procedure does include a 30-20 harvest control rule. I should note that although I showed a graphic yesterday showing that the relative biomass is estimated to have been below 30% in the past, it has never been estimated to be below 30% in the year in which the decisions were made. And so there is very much a 30-20 harvest control rule in effect that would linearly decrease the target rate of fishing intensity anytime the stock was below SB 30% down to a value of 0 for the directed fisheries at SB 20%.
Go ahead, Jeff.
Thank you for that answer. And then I would just ask that if we could get that policy in writing and maybe send it that direction. I've even heard the NIMS advisor to the U.S. commissioners cast a lot of doubt on this thing, and it's been said over and over again. And I think it's really important that they understand that we take conservation of this stock very, very seriously, and that when we do get below these thresholds, that we will take action at the commission. And so I just think it'd be helpful to reinforce that. So as they're dealing with abundance-based management regulations at the council, that they can take this into account. Thank you very much.
Next up is Chris Sporer, followed by Rick Koso.
Commissioners, my name is Chris Sporer. I'm from Canada, commercial fishing sector. Lots of things could be said here. I just want to respond to a few of the comments. There's seems to be a lot of comments about the agreement that was reached in 2019, and I just would like to remind everybody and make the point that yes, in 2019, Canada and the US reached a 4-year agreement that included specific allocations for area 2A and 2B, okay? That took a year to negotiate. If you look at the annual report from the 2018 annual meeting, it is quite specific there that the commissioners will be meeting to look at alternative ways to distribute the stock. So no one should be surprised. The commissioners met throughout the year. Okay, so there's meetings, and then it was discussed, and then at the 2019 annual meeting, both countries met with their delegations and provided to get advice and provide feedback. And then an agreement was reached in 2019, and it is clearly written in the 2019 annual meeting report what was agreed to. Like I say, it took a year to negotiate. At the same time, there was also an agreement to work on mortality and on bycatch, on monitoring and mitigating the bycatch. The commissioners explicitly looked at taking a step towards a more comprehensive and responsible management of the resource. And that was an agreement that the TCEY would include U26 mortalities and that both countries would be responsible for their collective U26 mortalities. Now, we've heard lots of discussion today, comments that that seem people maybe don't think that's such a good idea now, or we need to take more time to study it. Just want to make the comment. I've been coming to these meetings since 2000. Every single year we talk about bycatch. There's been numerous studies about bycatch and what it means to this area and that area and this area and that area. None of them are perfect. But if we we can't just say we're not going to do anything and say, let's study it again. That's my concern. We've been studying this to death. By not addressing this, it hurts the resource and it hurts all IPHC regulatory areas. And what this concerns me is what I'm hearing in a lot of the underlying the statements is, we don't want to pay for the U26 bycatch. We want Canada to pay for it. Everyone has the U26 discard mortalities. They're factored in and they need to be taken into account. They can't be ignored. Thank you.
Next.
Thank you. Next is Rick Koso, followed by Clay Duda.
Commissioners, my name is Rick Koso. I'm from ADAC, president of the ADAC Community Development Corporation. Today I'm going to be speaking on Area 4B. Don't try to balance the books by cutting 4B, and here's why. And I just want to give you some points on that. The number one, it may work on paper, but you can't force the halibut that live in 4B to move to other areas. Number two, tagging studies don't show significant movement of 4B halibut into other areas. Number three, 4B percentage shares of the coast-wide TCEY should be 4.8% based on the table 1, page 7 of the 2020 AM096.09. Recent stock distribution estimates by biological region based on modeling of all Pacific halibut captured by the FISS. Number four, 3.9% of the coast-wide TCEY shown in the default distribution tool as a result of reducing the exploration rate by 25% in 4B relative to other areas. Number five, if the books need to be balanced, 4B should be the last area to be asked to take a cut because we have given 25% up front already. And because in the real world, there is no biological benefit to other areas for cutting 4B. And just another note, ACDC also is a holder of a CQE in halibut and ADAC. And the bottom line, with the coast-wide TCEY of 31.9 million pounds, Area 4B TCEY should be no less than 1.24 million pounds for a FCEY of 1.06 million pounds. Thank you.
Thank you.
Thank you. Next is Clay Duda, followed by Travis Blount.
Thank you for your time. My name's Clay Duda. I'm a charter operator out of Homer, Area 3A. Some of the stuff I was going to talk about, these guys have already touched on it, so I'm going to try to be to the point. One of the hardest things for me is the uncertainty in regulations year to year. It makes it increasingly tough for the charter industry to maintain a viable business model. Changes in allocation affect our business models differently than they do the commercial sector, but I'm sure I don't really have to explain that to you. Excuse me. From my experience in 3A, our charter industry is sitting on the cusp of this no longer being a viable business model. This is one of the few, if only, entry-level fisheries left here in Alaska. I'm looking to get into that. I mean, I was on the cusp of buying a boat and a halibut permit this fall. And I mean, we were looking at a potential increase in allocation in 3A last year based on the research or, the science. And then this year we're looking at up to a 30% decrease. I mean, you're looking at whatever, I mean, 40, I don't want to inflate the numbers, but a 40 to 50% swing in a single year. Like it's really hard for me to justify a major investment based on such a wild card. You don't even know you're going to be able to do 1 year to the next. In the short term, I hope as it relates to 3A that we see our allocations at least stay to the status quo levels. I support the recommendations that are going to be put forward by our 3 representatives to kind of keep us around 1.7 million pounds for 2020, if I understand it right. And I think that, I mean, looking at our business, the charter business in 3A, that's pretty close to the bottom of the barrel for us. Yeah, I think that's pretty much everything I wanted to touch on. I'm happy to answer any questions.
Seeing no questions, thank you.
Thank you.
Thank you. Next up is Travis Blount, followed by Jerry Martin.
Thank you. I'll be brief. I appreciate your time and everyone's time. I'm a charter captain in Homer. I've been involved in the fishery since 1999 as a teenager working as a deckhand. Through that time, I have seen what has happened with the increasing regulations on us. I think that you're not going to find anyone with a vested interest in the halibut, the future of the halibut fishery, that doesn't have conservation close to their heart. The important aspect that I want to echo what Clay said is the variation in year to year. I am not a permit holder, and looking at trying to get into the business with the high level of uncertainty as to what we're going to be allowed to do in the next 3 to 5, 10 years makes it pretty precarious. There's a lot of new, from what I've learned being here, there's some protocols that have been implemented recently that don't have the time tested as far as sexing the fish and understanding, getting more exact details. I think that given that this is a new system, I would urge that status quo be strongly considered, given this is a new way of accumulating data. We're interested in the future of the fishery, but we also are asking for something pretty reasonable. 1.7 is a small slice of the pie, given our impact on the local economy. So thank you.
Thank you.
Thank you. Next to the microphone is Jerry Martin, followed by Garrett Elwood.
Good afternoon. Thank you for allowing public testimony here.
My name is Jerry Martin, and I'm owner of North Country Charters in Homer and have been for the last 40 years. So I've seen a lot of, witnessed all of the ups and downs of the quota and limited entry and the battles between the charters and the longliners. Brought us to a GHL, then limited entry for charter fisheries, and all the different ever-changing management tools that we've been living under, which currently is a catch-share plan, which is squeezing both of our sectors to the point of instability. So after sitting through all the data, the scientific data yesterday, I went home last night to try to digest that information. And I don't pretend to understand all the nuances of figuring out allocation. But I'm wondering, with all the science and data collection in the different areas, what it is about when the council can then arbitrarily take quota from one area or set of stakeholders and move it to another area and stakeholders. So how heavily does the commission weigh the science against their decisions? And if it is just that easy in these just drastic times, I respectfully request that some of this quota goes back to Area 3A to keep from gutting our industries further. Thank you.
Thank you. Next up is Garrett Elwood, and the last commenter for this morning is Greg, who didn't write his last name. So Garrett Elwood followed by Greg, and that's the end of the list for this morning.
Thank you, Commissioners. My name's Garrett Elwood. I'm Area 3 and Area 4 fisherman. My question is if Commissioners could maybe offer a little insight into the harvest recommendation for Area 3A last year that was represented a increase of about, what was it, 5%, when the weight per unit of effort for that area was down 8 and down 7 the year prior. Also, at that time, it seems that we had the knowledge that there was a significant gap in recruitment. So yeah, I'm just curious if you could offer an explanation as to why 3A went up last year after a decrease in weight per unit of effort.
Thank you. If I could ask Dr. Stewart to come up and address the choices that were made over the last 3 years. It's one of the last slides in your presentation. So this is near the end of Dr. Stewart's assessment presentation, and it's the slide that has the harvest choices that were made over the last 3 years.
Sorry, could you please repeat the question?
Yeah, so I'm just looking for some clarification as to how we ended up increasing the total removals for 3A for the 2019 harvest from 13.15 million in 2018 to the adopted recommendation was 13.82. After the previous 2 years, we've seen declines in weight per unit of effort.
We could have that slide. I believe it is near the end of my presentation. Has 2 comparison tables below this in the interim management procedure section. perhaps around slide 50 something. I can get there myself. This slide. So I can speak to the change in the reference TCEYs from 2018 through 2020, both in terms of the scale as well as in terms of the distribution. So each year we updated the information based on the results of the modeled survey in that year, which indicated a changing distribution over this time period. In addition, the stock assessment estimated a different level of relative fishing intensity. So where in 2018 we had no indication of incoming recruitments, and the forecasts were for a more rapidly declining stock with a larger effect of fishing, we had a relatively small overall reference TCEY. In 2019, with the first information available from the 2011 and 12 year classes, that provided a more uncertain but definitely more optimistic assessment result, which is why you see that result go from 31 million pounds at the coastwide level up to 40 million pounds. In addition, we had the results of the recently completed expansion that occurred in 2018 available for the 2019 stock assessment, which did not yet include all of the areas on the coast. It included everything but areas 3A and 3B. The big change from 2020, going back to a much lower TCEY in regulatory area 3A, also a lower total. The lower total is related to the effect of the new sex ratio information from the directed commercial catch, which has increased our estimate of the level of fishing intensity. And the change in distribution, which you see quite substantially there for regulatory area 3A between 2019 and 2020, is related to the survey results that we saw in 2019, primarily that large decrease in regulatory area 3A of 17%, the 032 weight per unit effort, relative to a 5% decrease at the coastwide level.
Thanks, Ian. I guess just more to be more precise as to what I'm inquiring about here is that it seems to me that there's been a deferred, that we've been waiting to hold off on the reductions when clearly we have a lowering spawning biomass. I'd just like to offer my commentary that although 3A has experienced declines in weight per unit of effort for the last 3 years, that the harvest in that area actually went up by 310,000 pounds. And in 3B, we experienced survey declines and we adopted the full cuts, totaling 1.3 million pounds. It's tough, you know. A lot of people talk about how they're at a cliff, and I don't really want to play that card. I just want to be treated fairly. Most of the recommended harvests that are circulating amongst the conference board on the U.S. side are suggesting that 3B take 100,000 pound increase after a 26% increase in weight per unit of effort and a 44% increase in commercial logbook data. And it's just hard not to feel like we end up bearing the burden of conservation for Region 3 in a disproportionate level. I guess I'd just further add that 100,000 pound increase for 3B is pennies when we talk about relating that percentage to the distribution and the estimated harvest that would be coming out of 3A. You know, I know that nobody thinks that this resource would, the condition of the resource would allow for an increase in an area such as 3B. However, given that we have taken the appropriate cuts, totaling 31% in just 2 years, it's a bitter pill to try to swallow, that we might only be, you know, gaining back a couple percent relative to what the distribution estimates are showing for 3B and 3A. And I guess I just finished with that. is that estimated distribution of the stock and spawning stock in 3B is 12.1%. We're currently harvesting 6.6% of coastwide total removals, where at 3A, estimated distribution is about 30.8%, and they're harvesting 35% of the coastwide removals. Thank you.
Thank you.
Next up is Greg, didn't leave his last name, for one comment from here in the room, and then there's one question online, and I'll preview that it's for Dr. Hicks.
Well, I'm going to be so brutal, I wanted to be anonymous. My name is Greg Elwood, and you just heard from my son, and I'm going to speak to the Area 3 conundrum, if you will. Sec here. We clearly have a situation where Area 3 is in a declining trend. Dr. Stewart has quite precisely forecasted these declines, warning us all of the gap in recruitment for the next 3 to 5 years. Despite the obvious fishery trends, lower weight per unit of effort over the 10-year time series, lower WPUE over the 10-year time series, lowering distribution estimates, 3A is refusing to accept the reality. Despite a 10% decrease in the survey weight per unit of effort from 2017 and 2018, the total removals from 3A increased 5%. I meant to say decrease, I think I said increase. The lower weight per unit of effort survey performance for 2019 in Area 3A should not surprise anybody. Region 3 is comprised of fish predominantly undersized 100 centimeters. Referencing tagging studies from Quinn, 1990, and Webster, 2013, we see high levels of immigration and emigration between areas 3A and 3B. The pit tagging studies conducted in 2003 through 2009 revealed that Region 3 is one biological stock where fish routinely move across an imaginary line off the west end of Kodiak. It cannot be stated strongly enough that this relationship between 3A and 3B is unique because it is the only region that shows strong level of migration to and from east and west, or mixing, if you will, of the two areas. The results of this year's annual survey and the trend over the last 5 years offer a visual assistant to this fact. Area 3A and 3B share 12%. My son just said this, so I'm kind of going over it quickly here. There are considerable difference in relative distribution estimates. 5-year distribution averages for 3A and 3B are 32.9 and 11.2% respectively. I would like to comment about the annual coastwide survey. It was down 5% for 2019. Some harvest areas might have been up or some down relative to last year's survey's coastwide stock estimate are still lower. Furthermore, there is little reason to think that this trend will be reversing itself anytime in the near future. We have a large gap in recruitment, which is almost certain to affect Area 3, Region 3, and eventually Area 2. I feel compelled to speak out in opposition to the eastern areas that continue to lobby and vote for quotas vastly above the estimated distribution, while expecting the more poorly represented areas of the west to pay the bill. 3B doesn't have a paid lobbyist. As a matter of fact, our family, my son Garrett and I, have been the only voice for 3B. We are not accepting a cut in 3B as it is not clear that we are not accepting a cut in 3B as it is very clear that 3A is not willing to take the cuts that the science clearly indicates that they should. Keep in mind that 3B and 3A share the same stock. We acknowledge the reality of a declining biomass. 3B has accepted the results of the survey and has taken 100% of the cuts in 17 and 18, resulting in a concurrent reduction of 31% in the previous 2 years. In the same year time frame, weight per unit of effort declined in 3A by 7%, by 8%, and finally this year's 17%. Take this in. 3A actually increased by 310,000 pounds over that time frame. If 3A were to take the cuts that were applied to 3A in 3B in the previous 2 years, this year's balancing the books would go away. If 3A were, we are not going to be the area that has been used to balance the books again for a system that won't even accept the best available science. Thank you.
Thank you.
Thank you, Mr. Chairman. That's all the comments from the local audience. On the web, I have a question from Mel Erickson for Dr. Hicks. The recommended fishing intensity of F43 would give the guided sports sector about 1.7 million pounds, resulting in severe restrictions. Does fishing at the current 2019 guided sport levels of 1.9 to 2 million pounds present a danger or high risk to the spawning biomass? In other words, could the guided sports sector fish at an intensity of F40 without harming the biomass?
Okay, yeah. Thanks for that. I guess I would start off saying that the MSC is looking at the management procedure from a strategic point of view, and that's where the 43% comes from. There's also a tactical decision-making process, and that's what the commission does at the annual meeting, where they try to make those annual decisions for this. So the 43% is a recommendation coastwide, all sectors. And from the MSC results, I really can't answer what would happen if the sports sector or recreational sector operated at a different fishing intensity than other sectors. However, the decision table that Dr. Stewart has presented might be the place to go to really look at what the risks are to spawning biomass over the next 3 years if harvest was increased by certain amounts. And I imagine if you, maybe you can use the mortality tool online as well to take a look at what are harvest levels, and adding a couple million pounds or however many more pounds might have a risk to the probabilities on the spawning biomass in the decision table.
Thank you, Dr. Hicks. And does that conclude public comment?
Yes, Mr. Chairman, that concludes the public comment for this session. Thank you, Mr. Chairman. So as a reminder for logistics here of the meeting, we have a hard stop here at 12:30, and they're going to rearrange this room for the conference board, which will convene here this afternoon. The commission will continue its meeting in the room next door, and the processor advisory board will start its meeting this afternoon downstairs. So we're not getting to agenda item 11, which is regulatory proposals for 2020 at this morning's session. I'll just preview the process for regulatory proposals that we're following during the course of the meeting. I will be able to present this to the commission this afternoon in their meeting, and I'll tell the conference board and the processor advisory board that I'm happy to make the same presentation to you at any point during your proceedings as you desire. The presentation this morning is an introduction to the regulatory proposals under consideration at this year's meeting. There is time in the agenda both tomorrow morning and Thursday morning for the commission to consider the regulatory proposals and have further discussion, further questions. And Thursday afternoon, after the conference board and the processor advisory board reports have been presented to the commission, is the first time we will ask the secretary will ask the commission to make decisions on the regulatory proposals. As we've done in the last couple of years, we would like to take commission action on as many of the regulatory proposals as we can on Thursday afternoon, which clears the decks for Friday morning to get to the fishery limits and the fishing period dates, which is what everybody's waiting for on Friday morning. I'll just note that there are not very many regulatory proposals for this year. There are 5 from the Secretariat, 2 from the contracting parties, and 2 from stakeholders. They're all online. You can see them. They're relatively straightforward. The ones from the Secretariat include the normal first 3, which are the fishery limits. That's the place where the fishery limit decisions make their way into regulation. The second one has to do with the fishing period, and so that's where we talk about the season dates. And the third one is minor amendments, which we offer pretty much every year because we find something that needs to be changed. There's 2 additional ones that have to do with vessel clearance in IPHC Regulatory Area 4, as well as the IPHC closed area. And those are the Secretariat proposals. The 2 proposals from the contracting parties, one are both from the National Marine Fisheries Service. The first one has to do with charter management measures for regulatory areas 2C and 3A. And the second one has to do with the delimitation of subarea 2A1 in IPHC regulatory area 2A. And then the 2 stakeholder proposals have to do with regulations concerning the recreational fisheries in Alaska. So I'll invite your attention to those. They're all posted online. Again, I'll make the presentation for the commission this afternoon whenever it fits into their program, and I'll offer it to the conference board and processor advisory board at their pleasure. And there's times in the agenda between now and Thursday afternoon when we will ask the commission to start making some decisions regarding the regulatory proposals. So that's all we'll do for now because we're going to run out of time this morning. If you have any questions about regulatory proposals, please see me at any point during the proceedings. Thank you.
All right, thank you. We'll break for lunch now and reconvene next door at 1:30.
So just a reminder, please be out within 5 minutes to assist us with the room flip. So take all of your belongings and head on out within the next 5 minutes. Thank you.